So, we all want medical treatments that actually work, don't we? That feeling when a doctor just gets what your body needs. And I think, when you're talking about neurodegenerative diseases where precision can make such a difference, well, that desire for a tailored treatment is even stronger. Today, we're doing a deep dive into some really groundbreaking research. It's bringing that future closer, particularly for folks living with Parkinson's disease. Now, as many of you probably know, Parkinson's is the second most common neurodegenerative disease out there. And the numbers are, unfortunately, growing pretty rapidly. It mainly hits the dopamine-producing neurons, in specific brain area. And that leads to those really challenging symptoms, tremor, stiffness.
Right, and slowed movement balance issues, too. It's tough. And for decades, the main medication has been Levodopa. It's really the cornerstone of dopamine replacement therapy. And look, for most patients, it does bring significant relief.
Okay, but here's the interesting part, the real question. If Levodopa is generally effective, why doesn't it work equally well for everyone? For some people, the results are just, limited, frustratingly so. And until now, it wasn't really clear why. So what's actually going on? What's the breakthrough here?
Yeah, that's exactly the question scientists at Simon Fraser University, SFU, and Sweden's Karolinska Institute wanted to answer. They've been using some pretty incredible advanced brain imaging tech to get at why levodopa has these varied effects.
And that tech is magnetoencephalography.
Nice.
Sounds really cutting edge. Can you maybe explain a bit more, like what is MEG? How does it give us this real-time picture?
Absolutely. So MEG, it's non-invasive, which is great. Think of it like a super sensitive microphone, but for the magnetic fields your brain produces. These fields come from the brain's natural electrical signals. And the crucial thing is it lets researchers watch brain activity basically as it happens. Very high temporal resolution. You're seeing the brain work in real time.
Okay, wow. So with this super-powered view, what did the researchers, people like Alex Wiseman at SFU, actually find? What did they uncover about why levodopa isn't quite this one-size-fits-all drug? What was the key discovery?
Well, the key thing they found, using a new way to analyze the MEG data, comes down to what they call off-target effects of the drug. So imagine levodopa is like a key meant for a specific lock in the brain, the one to help dopamine function.
Right.
What they discovered is that sometimes that key doesn't just open the right door, it can also sort of jiggle the locks on other doors, activate brain regions it wasn't necessarily intended to.
Ah, okay. So these off-target activations, are they stopping the drug from working altogether or just interfering?
Interfering is the right word, yeah. It's crucial to understand people experiencing these off-target effects do still get some benefit from the drug, just not as much as others might. These unintended activations seem to kind of get in the way of levodopa's main helpful effects, makes it less effective for those individuals.
That seems like a huge piece of the puzzle. So if we can pinpoint these off-target effects, understand them, what's the ultimate goal here? How does this knowledge lead us towards, truly personalized Parkinson's treatments, treatments tailored to my specific brain response?
Exactly. That's the exciting part. What's really fascinating is how this finding points directly towards personalized Parkinson's prescriptions. If we can understand exactly how levodopa hits an individual's brain signals and spot these off-target effects with something like MEG, well, then clinicians could potentially really fine-tune how they prescribe the drug, maybe adjust the dosage, maybe switch to a different medication earlier and more effectively. Like Alex Wiseman said, the more we can personalize that approach, make it more expedient, make it a bit more specific to that person, the better. It's really about moving from a broad approach to something much more precise.
And does this have potential beyond just Parkinson's? I mean, it feels like this understanding, this method, could it apply elsewhere? What about other neurological conditions or even other types of drugs affecting the brain?
Oh, absolutely. You're right on track there. The implications are potentially huge. This new analysis method that Wiseman and his colleagues developed, it's not strictly limited to Parkinson's or levodopa. It could, in theory, be used to study any medication that influences brain signaling. Think about epilepsy, depression, stroke recovery. The list goes on. It opens up so many possibilities for optimizing treatments across the board. Imagine applying this kind of precision, everywhere.
And is there a specific place driving this, a reason this work is happening where it is?
There is, yeah. It's probably worth mentioning the specific resource they have. SFU's Image Tech Lab, which is located at Surrey Memorial Hospital, actually has the only MEG facility in Western Canada. So that unique setup really allows them to spearhead this kind of research.
That's fantastic. So, okay, what's next then? How do they take this from the lab, from this high-tech MEG discovery, and make it something that can actually help more patients more widely.
Right. That's always the critical step, clinical translation. The team's plan is, first, to apply this new approach to a larger group of patients, validate the findings, and really importantly, they're looking at how to translate this to more accessible brain imaging methods, specifically electroencephalogram or EEG.
EEG, okay, for listeners maybe not as familiar, how is EEG different from MEG, and why is looking at EEG so important for, getting this out there?
That's a great point. So, while MEG measures those tiny magnetic fields, EEG measures the brain's electrical activity directly, usually with electrodes on the scalp. It works on a similar principle looking at brain signals, but the technology itself, EEG, is much more widely available. It's less expensive, more common in hospitals and clinics. So if they can adapt their analysis for EEG, it means the benefits of this discovery could potentially reach way more people. A much more diverse patient population, clinics everywhere, not just places with these rare, expensive MEG machines. And, this all raises a really important question for you, the listener, to think about. Just imagine a future where all drug therapies, or at least many of them, could be precisely mapped to your individual brain's response. How might that shift, moving from generalized medicine to truly individualized brain-specific treatments? How might that revolutionize healthcare? Making every prescription potentially a precision prescription, it really could fundamentally change how we approach medical care. And by the way, these specific findings we've been discussing were published in the journal Movement Disorders.